Evening and welcome to the third of the series of Prof Talks with Professor Andrew Shaw and me, Jonathan Snicker. It's a great pleasure to welcome you this evening. We're just waiting for everyone to join before I introduce Andrew to the group. And whilst we're waiting, a brief survey will appear on your screen. If you take a few seconds to complete that, please. And this is for you to describe yourself as you were viewing this webinar. Thank you very much. Professor Shaw, welcome. How are you this evening?
Enjoying the sunshine.
Good. Well, would you mind, Andrew, as is customary, introducing yourself and explaining in brief how the technology works and your vision for the test?
Yes, of course. So everyone, hopefully, who is joining us, again, welcome back. Those who are joining us for the first time, we've been looking at a new way of characterizing the antibody response in long COVID patients, which enables us to get a view about how the long COVID community is patient community is potentially suffering from persistent virus. So without this becoming an immunology lecture, I can share a couple of slides with you. So this is the work we're doing, and I'm going to talk briefly around the hypothesis. So we are in the position where we think that once you've had your long COVID infection or acute infection happens, you've had the virus inside you, your acute phase rises, all your immunology gets rid of the fever, gets rid of the sick cells, and the virus can be in your lungs, it can be almost everywhere around your body. And over a period of time, those symptoms don't disappear. Why don't they disappear? Well, one of the theories is persistent virus hypothesis, maybe even persistent spike protein. And that means there are places, reservoirs in your body, around your lungs, the digestive tract, your heart, your brain, your liver, etc., where the virus is hanging around. These viral reservoirs might cause inflammation, and we're now beginning to see that from a new cohort we're studying at the moment. They can cause endothelial dysfunction, they could cause clotting, gut dysfunction for sure, and brain fog. And these post-viral sequelae, post-acute sequelae just don't disappear. And for some people, we now know this journey is 5 years long. And even if we were to find a cure, how quickly do people recover, which is a really interesting question. So what we've learned at the top here is that if you measure somebody's antibody spectrum from a group of people not suffering from long COVID, you get two measures here in our test. One is how many antibodies they got for each of the variants. So 14 variants now, 15 variants across the bottom. We added XEC in the last cohort. You'll see the new spectrum shortly. And that tells you the concentration. So if you've got the virus sitting in your body somewhere, you need enough antibodies to go and find the virus and then to stick to it and trigger the clearance process. But if the sticking isn't any good, then this quality level is bad. So this is good quality antibodies. 60% of the antibodies are high quality. And that means if it sticks to the virus, it will stay there for more than a couple of hours. And when it does, that means the clearance processes in the body will then work. In about 65% of long COVID patients, the antibody spectrum in the two measures I've just mentioned looks like this. So you can see the concentration is very patchy, and for some variants, there's hardly any antibody concentration at all. So going back to my idea that you've got to be able to find the virus, if you don't have enough antibodies, in this case for this patient around Omicron BA1, BA2, for example, then the virus just won't be cleared. And as you can see, the antibody quality is non-existent for the later variants. So if this patient is suffering from long COVID associated with, for example, an Omicron variant, then you can see why they haven't cleared the virus. It's hanging around, there's a persistent virus. 10% of the patients have a very different response where they have very high antibody levels, around 140 milligrams per liter. So in the case of that's nearly 1.4 times that, 10 times that. But the antibody quality is really good, all of those antibodies. And this is possibly because this is an autoimmune response. This is a hyperimmune response, as I've called it, because we don't know what's really causing it. But for whatever reason, these antibodies are there, very high concentration, and they don't go. They don't drift over time. For 25% of the cohort, their spectra look normal. I can't tell a difference in the antibody spectrum at all. So there are three types of long COVID patients. Now, some that would be eligible for some sort of repair of their antibody spectrum, and some would be eligible immediately for treatment of their autoimmune hyperimmune position. So as I mentioned, we've now got...
Andrew, may I stop you there? Because I think we'll come back to the US cohort in a second. I just want to run through a little bit of the housekeeping. Over the next 30 minutes, we are going to be answering your questions, as well as Andrew explaining this test, which we accept is complex, and we'll unpack it. Andrew will unpack it during the course of this session. Please put your questions in the Q&A section of the webinar. You'll see it at the bottom of your screen, not in the chat section. We can't pick it up in the chat section. This meeting will be recorded, and it will shortly be available on YouTube. And Andrew's actually, I think you're busy with webinars this month. If I remember rightly, you've got another 3 to do. He'll be in conversation with Dr. Ben Sinclair, the founder of Dr. Finley's, I think next week. This webinar is about the science behind the COVID antibody spectrum test and some of the treatments the clinicians are using. It does not constitute medical advice. Please speak to your clinician about the Attomarker test, or provide a link to your clinician to the website. The meeting will be recorded, and it will be available on YouTube shortly. Sorry, that was a rather sort of boring introduction. Now Andrew has introduced the test, and I think one of the questions that we had, which links to the screen that he just had visible. Is it possible, Andrew, that someone can be both autoimmune and immune compromised at the same time by the results of this test?
We may have just seen this for the first time, which is really interesting from the American cohort. So this is the US cohort. We've just had 50 tests over from America. This has got the new XCC variant on it. And you can see this patient here has enormous antibody levels, more than 200 milligrams per liter. I think this patient's more like 800 milligrams per liter. This is the biggest autoimmune response we've seen. And all of them high quality antibodies, right? So the antibody level quality is falling a bit down at XCC, which worries me gently. But this is absolutely not just hyperimmune, but maybe MAGA immune, I suppose. But here is an example of a patient that has high antibody levels here for alpha at 200 milligrams per liter, low antibody levels here for XCC, and look at the antibody quality. They're all getting quite low here. I think there's a risk that this patient here is suffering some autoimmune response and will have symptoms associated with that. But now, if they were to get an infection from one of the new circulating variant, XCC or the Nimbus variant around at the moment, their antibody quality is quite poor. So now there's a real possibility that this patient might have both hyperimmune and immunocompromised responses, and that then becomes a serious problem.
Thank you. Andrew, and that then links to, thank you for your questions. We're getting questions coming in now. What are the treatment options clinical partners for Attomarker are choosing, both with respect to the hyper or hypoimmune, and I guess this kind of combination, which is going to be even more complex. Could you speak to that, please?
So our clinical partners are looking at the antibody test as the part of the clinical workup for a patient. And if a patient were to be in this category here, there are some options around how you might treat this. So could we, for example, add a monoclonal antibody that works across the spectrum to produce effectively a synthetic antibody spectrum and use that to clear the virus? And so Cavigal or Sipavibart, to use its technical term, is the AstraZeneca antibody of choice out there at the moment. It has been approved for use in a number of countries, including the EU and Canada, not yet in the UK, not yet in the US. And there is an option now to import Sipavibart into the UK, and there are, I know, nearly 100 patients waiting in the partner clinics to get the intervention. What would happen then? You go to the clinic, they would give you the antibody as an injection intramuscularly. The protocol that the clinicians are exploring is 3 shots separated by about 40 days. That means the antibody concentration from the Cipavibart will begin to rise in your blood quickly, and this would then level off, and you get a spectrum that look much more like this normal response. And the reason for 3 shots is it's got to perfuse around the body. It's got to get out of the bloodstream into your gut, into your head, into your nerves, even into your lungs. And that would then result in a potential binding, sticking for long enough, and then a clearance process. These patients down here that are hyperimmune, that's almost a field of medicine of its own around rheumatology, autoimmune diseases. There are a number of ways forward here, including immunosuppression, immunomodulation. So drugs like methotrexate could be used to switch off your antibody production, and you should get some benefit from that. The other 25% need to be treated symptomatically. What's the medical analysis of their symptoms and what might be targeted? So the clinical partners are looking at all 3, so you won't get abandoned. All 3 subgroups will be treated, and interventions are possible clinically for these. The hyperimmune straight away, and the immunocompromised when Cipavibart becomes available.
We're having further questions about treatment. Thank you, Andrew. And one question is around apheresis, which I know is available on the NHS in about 8 centres across the UK, but for other conditions, cancer, renal conditions, and so forth. Is apheresis being used? Are clinicians using apheresis in respect of these symptoms?
So apheresis in one of our partner clinics in Europe is being used at the moment, and is now going to be considered by Recognition Health in London. What happens here is that all of the components of your blood are removed except for the cells and the platelets. So all of the proteins in your blood are removed, which means if you're hyperimmune, all of these antibody concentrations should go down to something really rather low. And what they do then is replace that fluid with something else. And the replacement fluid could be salt water, it could be salt water with proteins in it, it could even be previously cleaned plasma. And we know from the clinic in Cyprus that patients get some effect from that effectively quite quickly. But for most patients, the problems return. Now, why is that? It's possible that if you're hyperimmune, then the antibody production just gets switched on massively fast, and that means antibodies are then made, hyperimmune antibodies are remade, and the system just resets. So interesting idea that you do apheresis, clear out the blood plasma proteins, then do the intervention around immunomodulation, switch off the antibody production, and so you could stop that returning whilst all the but you could leave the patient very vulnerable to all sorts of infections without antibodies. You could contemplate putting the Cipavibart in at that point, but what would it do? Probably nothing. For this patient here, you would clear out the blood, including these poor antibodies, and if there was any spike protein in the blood or any persistent virus in the blood, then that would go, and we could look for that in the effluent that comes out, in the liquid that comes out. And then you could add the Cipavibart into effectively what would be a clean bloodstream. That might be effective because it would stop the Cipavibart being mopped up in the blood and allow it to push out around the body better to get to those parts of the body where the virus may be hiding. So I think it's unknown, and in combination with the antibody therapies, it's definitely unknown. And I think some clinicians are now weighing this up as a really interesting proposition.
Yes, and it should be emphasized this is emerging science, and Andrew speaking from that kind of perspective. The one question which I will partially answer, and then you can correct me if I'm wrong, Andrew, which is always fun. Could you summarize the current value of these diagnostics, if any, with respect to existing treatments? My understanding from what you've just said is that these are signposts, and obviously a treatment modality for someone who's hyperimmune compared to hypoimmune has to be different, because you don't want to add antibodies to someone with a hyperimmune response or to cause them to create even more antibodies. Is that correct?
Yeah, I think we've now tested more than 450 patients. And actually, I think long COVID is now a bit of a misnomer for these hypoimmune, these immunocompromised patients. They are not the same as the hyperimmune patients. And so why would you treat these the same way? There are trials, for example, around the use of remdesivir, which is an antiviral. That might be useful for this patient, 65% of the cohort. Why would it be interesting here? Now, that would go against the hypothesis I proposed at the beginning. It might work. And what would it do for this 25% here? We can't tell. But it looks to me like from the antibody spectrum, long COVID now divides pretty directly into three. And I think we have reasonable evidence to suggest that all subsequent interventions should be considered against this or these three subcohorts.
Thank you. That's very helpful, Andrew, and actually good to be somewhat right in my response. Here's a very technical question. Any thoughts on immunomodulators to reduce hyperimmune response, for example, LDN and low-dose rapamycin? Have any Attomarker patients with a hyperimmune response tried low-dose rapamycin? I should add, Attomarker doesn't have any patients. The clinicians have patients, and reported back on how they've got on. Could you speak to that? Have you got any information on that?
So one of the patients in the American cohort had been on low-dose rapamycin for over a year and reported nothing of value from that. So that drug in particular for immunomodulation hasn't been overly successful. There are other drugs like methotrexate, which is known to suppress antibody production for things like rheumatoid arthritis. That would be interesting, but again, you need a rheumatologist and a clinician to think about that. I think there's a reasonable set of steroidal and non-steroidal immunomodulators. And I think that's just about experience from the individual clinician and what they would recommend and how they would recommend dosing it. From that point of view, the hyperimmune patients, possibly autoimmune patients, have treatment options almost immediately and should set about doing it, because I think a prolonged exposure to a hyperimmune autoimmune response is not going to be good in terms of persistent symptoms and potentially even damage.
Thank you, Andrew. Let's pause just to take stock for a second. Another survey will appear on your screen in a moment or so from the redoubtable Emily. Very simple answer. Have you taken an Attomarker COVID antibody spectrum test? Thank you very much. Right, we'll move on. There is some absolutely excellent questions coming in. I don't know if we can answer them all. They've suddenly been hidden by that poll. Oh, there they're back again. Now, there was a question around price, which I know is a contentious area, which is also related to off-license use. So let's ask the question about price. It's a difficult one to answer, Andrew. It's more of opinion than an answer. I've been able to source Sipavibart in the UK for September 2025. Wow. At a cost of 4,975 for a single dose, double wow, excluding injection, consultation, and follow-up fees. Is this pricing expected and is it considered fair?
Interesting question. We are in the process of trying to get an import license for Sipavibart from another source, which we hope will be here shortly. We expect the price to be a bit less than that, but we haven't added the full consultation fees in. I think the answer is going to be somewhere between £3,000 and £6,000 per dose, because that will depend on how it's shipped, who's shipping, what the costs are going to be. But the value is the important bit. If you can get hold of it now and the bird in the hand is worth two in the bush, if you're immunocompromised and you know your antibody spectrum is poor, the right thing to do is to take it when it's available. The value of having the drug option is high. And I have an anxiety that it may not be around for a long time. Sipavibart has now become vulnerable to a mutation, F456L to be precise, which means it's not working very well against Nimbus and maybe arguably not as well against XEC. If that mutation is a problem, it's possible AstraZeneca might consider withdrawing it. So I'm concerned that although it has real application for long COVID, I really want to get to large cohort tested so we can see whether repairing this spectrum can be made by sipavibart. But if AstraZeneca stop producing it, then nobody gets to try it. I'm chatting directly to the team in Washington, D.C. around the future for Cavigal sipavibart, and I hope they won't withdraw it soon. And arguably, I'd like them to repurpose it away from the license, which is for the treatment of the acute phase, and that's not going to work right potentially against Nimbus, and keep it repurpose it for the long COVID application. We just need data to suggest that it's promising, enough people recovering, and then hopefully AstraZeneca might be interested in trying a new repurposing.
Thank you, Andrew. Another therapy that's been discussed is whole virus vaccine, Novavax. The question is, are there any updates on the Novavax trial or availability of the updated vaccine?
So the data Novavax are publishing around protection for XEC and Nimbus is good. The antibody levels are waning a bit. In other words, the antibody levels produced by the virus, by the vaccine, are a bit lower towards the later variants, but they think it works. And so the Novavax is a protein vaccine, which I like. It should stay on your arm better because of the way the vaccine is made. And it should then enable you to repair naturally this spectrum rather than filling it with synthetic antibody. It should let you repair it. I was chatting quite well to Novavax, which I think is actually being manufactured by Sanofi at the moment. That has changed somewhat, and so I don't know what the availability is going to be. If they bring it out again in the autumn to coincide with the flu season, then we would have the opportunity to put intervention around Novavax. We're about to publish a paper, pull together a paper for reporting the interventions we've done. I think there are 3 vaccine interventions that show success. So again, I'm hoping that Novavax might be available again in the autumn, and we can try antibody spectrum directed interventions.
So, as I understand it, Andrew, about 450 people or over 450 people have taken the test. Obviously, access to the therapies itself is limited, but is it around 6 or 7 people have gone through some kind of treatment or therapy with clinicians? And are there any reports on outcomes there?
6 people we know of have been through therapy that has been effective. Either the antibodies, sotrovimab or Evusheld, worked well on 2 people with early variants. They work very well against the early variants and not at all against the later variants. This would be a reasonable spectrum for sotrovimab. That worked well if you have long COVID associated with the beginning of the pandemic. That would work. We've intervened with 3 people. One of our partner clinics has intervened with 3 people with Novavax. That repaired the antibody spectrum, and that worked as well. What does work mean is really important to understand, because recovery may not be immediate. Recovery may be quite slow. Some people have been suffering for long COVID for 5 years. Doesn't follow they're going to get out of their bed one morning after intervention and just feel better. That's going to be a journey. But we've seen things like heart rate variability improve quite quickly, and then migraine symptoms improve quickly. So I think there's reasonable. 6 is not a cohort. 6 is not significant. There is one person who had Evusheld, but their antibody spectrum said they had good antibodies to the early variant, and their persistent virus was probably Omicron. So that's a negative. They were given the vaccine, the antibody, and it didn't work. And some people in the PEMGARDA trial have also been reporting transient effects, maybe no variation at all. They're not they're not selected by the antibody spectrum. And we're hoping that the sipavibart trial in Florida with Nancy Clement, no, I can't remember her surname, will be subject to at least a post-hoc analysis of the patients with the antibody spectrum.
That's at Nova University in South Florida, isn't it?
Yeah, Nancy Klimas. Yeah.
Great. Thank you, Andrew. Well, as is customary, I'm now going to put you into a university challenge and ask you rapid fire questions. If you would keep your answers succinct, but the usual high quality. I'm going to answer the first question for you, which is, will Anttomarker be interested in running tests before and after my sipavibart treatment for data collection purposes? Yes, please. would you ask your clinician who's managing that for you to contact Attomarker? info@attomarker.com would work, and other email addresses appear at the end of this. All right, are you ready?
Yes.
Very good. Okay, let's go to a subset of patients worsen with monoclonal antibodies. Do you think this could be in the autoimmune MCAS subset? Is there any way to predict this? You partially answered that, but brief answer, please.
Anybody in this 35% here may not do any better with antibodies and may get worse. We don't know what the cause of the autoimmunity is. If you add an antibody that's already sitting at 140 and you put another 140 on top, which is what might be possible from the drug, you're going to make it worse. It'll double the antibody concentration. It could double the hyperimmune response. So yeah, I think when this paper is published, I think all clinicians need to consider our subcohort of 3 here before giving drugs.
Yes, and this gives rise to one of the issues. Hyperimmune patients are very different, but their symptoms might be similar. And in medicine, one of the key problems is there's about 4,000 human diseases, but only about 400 symptoms. So this is why these kind of advanced diagnostics are going to be increasingly important, we think, across disease areas. Correct me if I'm wrong, Andrew.
That's spot on. That's the vision for Attomarker. I think we're seeing just this point. Symptoms don't tell the difference between these two. But when you do some multiplex clever science, you can see some things happening.
Great. In your previous webinar, Andrew, with Dr. Sinclair, you mentioned that if you have observed IgG4 is elevated in long COVID patients, that you have observed this. Have you observed this specifically in hyperimmune profiles? Very interesting question. Is there a good COVID IgG4 test that is commercially available?
Commercially available from us, but as a research test. So your clinician would have to request it directly. We haven't got the UK CA mark for that yet. We're working on that. We need a calibrant for IgG4. That could be available shortly. I like it because if IgG4 levels are high here, because there's exposure to the variant or exposure to the spike protein, then you would see the IgG4 level fall if we cleared the virus. And I think that would happen quicker than, say, symptom recovery. And so there's an interesting biochemical test of before and afterwards. Did something change? Was there a persistent virus? Was there a persistent spike protein? It's now gone. That would be a really interesting measure. IgG4 in the hyperimmune group. Now, I'm stretching my memory here, but I can't remember off the top of my head whether we have sufficient data around the differential analysis. I'd have to check that. Maybe at the next Prof Talk, I'll come back with that.
Great. Thank you, Andrew. I'm going to go about 5 minutes over time because the questions are so good here. Whilst this is a long COVID webinar, I have ME CFS as well. So I'm assuming the tests would not repair, but help with that too. Is that the case? Will that help people with ME CFS? Or is that a separate test?
I think it's a separate test, but I think the mechanism is the same. So ME is possibly persistent Epstein-Barr virus, maybe herpes virus, which we probably all had at some point in our life and all cleared. And for some people, that hasn't happened. I'm writing a grant with Prof David Strain here at the university where we can invent a similar test looking at the antigens for Epstein-Barr virus and herpes virus and see whether you've made good quality antibodies or bad quality antibodies. And that would then point to a diagnostic for ME. There are 3 companies from memory that are working on vaccines for EBV. So again, that might be a route out. If you are already struggling with persistent EBV and add in persistent SARS-CoV-2, I think that would probably be a worsening effect. And we could certainly tell the difference. We could certainly show that the persistent SARS-CoV-2 was present in your body from your spectrum. Does that mean you're just making bad antibodies all the time or just unlucky? I don't know. We would have to check that. So we can certainly now look at what you've got with SARS-CoV-2.
Great. Thank you, Andrew. So now the questions are going to become even more complex, which just shows how educated, self-educated the long COVID community and indeed the ME/CFS community is. Before we do that, there's one final poll that will appear on your screens. Give you a chance to get your brain in gear, Andrew. Final poll. After hearing our webinar today, would you consider purchasing an Attomarker COVID antibody spectrum test? Yes, no, or maybe. Thank you for answering that. All right, here we go. Are you ready? Could you speak to the limitations of monoclonal antibodies in reaching viral reservoirs embedded in tissues, particularly where there may be ongoing replication in sites like the gut, lungs, or CNS? Wouldn't additional immune stimulation, such as with interferons, be needed to help the immune system clear infected cells? That's part one of the question.
So yes, I think different viral reservoirs are going to be harder to reach. We know from the Evershed intervention that the gut actually is very well supplied by the blood, and gut function improved quite quickly for the Evershed patient. We also know for the Evershed patient it got through the blood-brain barrier, so we know that the antibodies can reach the brain. But the reason for the triple dose is to ensure that the antibodies perfuse around the body at high enough concentration. I think it's quite important the blood is cleaned up first. Once the blood is cleaned, then you go into the gut and that's cleaned up, and then you can push harder into places like the brain. So accessing the different reservoirs is the reason for the triple dose in the protocol that we're experimenting, we're suggesting to our colleagues, our clinical colleagues. Other doctors are trying it differently. So the PAMGARDA trial and the trial in South Florida is just one shot.
Yeah. Thank you for answering that, Andrew, and thank you everyone for answering the question. 57% said they would consider Attomarker test, 34% said maybe. In the next webinar, we should probably ask the question whether you would consider it if it was available on the NHS. Okay, second part. Do you see a role for combination therapy, e.g. Paxlovid to halt intracellular replication, PEG IFN lambda to stimulate clearance from epithelial endothelial reservoirs, with monoclonal antibodies to neutralize the virus? I hope you're taking notes in the blood and prevent reseeding. In more complex cases like CNS or immune cell involvement, could interferon alpha or high dose monoclonals with CNS penetration, e.g. Evusheld, you've answered this, be useful?
Paxlovid was given alongside Evershed, and the reason for that was I thought it was possible that the virus might be disturbed by the penetration of the antibodies. If that were then, if the virus would then find an ACE2 receptor, it should then start replicating. It was important it was Paxlovid because it's well targeted at the mechanism for messenger RNA DNA transfer, as opposed to molnupiravir, which for me is a randomizing of the viral DNA, which I didn't like in terms of not being precise. So yeah, I think Paxlovid on top of the monoclonals would be interesting. All the other immunomodulators are very interesting to try and encourage this reservoir access. I don't know scientifically which of those would be better, and you would have to defer to the clinician at that point. That's a serious combination of some fairly serious drugs.
Mm. Yeah, and there's such good questions here, Andrew. It's impossible to ask you them all and to answer them all. So I think we're going to collate them and attempt to respond in the next seminar. There's a mention of ashwagandha. We weren't aware as a team of ashwagandha as a therapy for long COVID. It's been used, we discovered today, by a famous sportsman in Canada. It's part of Ayurvedic medicine. It's a powerful herb. We can't really speak to that, Andrew, unless you want to add anything on the issue of herbal medicine more generally.
Many of our drugs have come from herbal preparations, aspirin being one of them. What we haven't done very well is to look at whether the combinations inside herbs are better than individual molecules. So what we've done is taken the good stuff out, thought that's one molecule is responsible for all of the interactions. I had visited Hong Kong Institute of Science and Technology and Hong Kong University when I was an international tutor, and they were looking at whether combinations are better. I haven't heard of any clinical trial that says we've looked at other molecules in herbs that work synergistically well together or more effectively. I don't see why it shouldn't. We've just been talking about combinations of immunomodulators, and that's a mixture, a complex mixture of drugs, as is a herb in some way. I think it's
I'll read out some comments because they're quite instructive. Why is this testing not available on the NHS? What are their hesitations in using tests like this to guide management? Or is there a whole set of issues around trials and procurement? What immune modulation drugs can help? I've been dismissed by rheumatologists as with no obvious autoimmune disease. Would Andrew, you offer to send that test result to the rheumatologist? What would be the best thing for this person to do?
If you had this test result, I think that could trigger a whole series of other autoimmune investigations, but I think that's a very good indication of this antibody level being high. You've got to ask why it's high, what the mechanism of the autoimmunity is, but I would certainly be happy to even discuss with the rheumatologist what the implications of the test is. It's not well known, the test, amongst all clinicians, and that's in part why it's not in the NHS. As ever, I think if we were to run a big interventional study and prove that it worked, then the NHS would adopt it. But that's a long journey. It could take several years.
Yeah. Well, I'm afraid, everyone, we're running out of time, and there's a lot of questions. I must apologize that we've been unable to answer in the time we have available, but we'll make a note of them and get back to you or find ways of answering them. One final question, which we again discussed today, Andrew. In your previous webinar with Dr. Sinclair, you mentioned T-cell assays. Which T-cell assays that are commercially available would you recommend, and could results influence treatment or be for info only? Or could T-cell assays be used alongside an Attomarker panel to maximize information?
There's a T-cell assay, activation assay run by one of our partner companies, Immunoserve. That then tells you whether your T-cells are able to make a good response to spike protein. And I think if you have recovered from the acute infection, your T-cells are probably okay. If it's been a really long journey, then to know that your T-cells are working would be useful too.
Great, thank you, Andrew. I think we'll close it there. Andrew, do you have anything by the way of closing remarks before I close the session entirely?
I really hope that we are lucky with the MHRA license for importing Sipavibart. We've located it through some partners now, and I hope that we can get some into the country quickly. To have the first 100 patients run through the intervention would be enough to stop it. If I'm not right, if this measure is not useful, then with 100 patients we will know that. If it is right, 100 patients is the beginning of a number that would begin to convince AstraZeneca and the government. So it's a really exciting summer ahead of us if we can get the molecules in. If we can find partners in Canada, if we can find partners in Europe where it's available, I would like to move on that really quickly.
Great. Well, thank you so much, Andrew. Thank you, Emily and Steve. Perhaps you can show yourselves to be thanked for supporting us in this webinar. Andrew will appear with alongside Dr. Ben Sinclair, the founder of Dr. Finley's, on the 18th of July next week, which will be his next appearance online. So it just remains for me to thank you. I hope you're doing okay. I hope this has been helpful for you, and we look forward to seeing you at the next Prof Talks webinar. Thank you and good night.